Okay, so going on, clonal propagation of elite plants, and then in bracket it's written shoot tips, axillary buds, and meristem culture. No, that is how they wrote it in your syllabus. Okay, the word clonal propagation. Don't get confused if you see the word micro propagation also in your exam. Okay, they both mean the same thing. Let me repeat. You are used to seeing the word clonal propagation, okay, in your syllabus. But if you see the word micro propagation also, it means we are talking about clones only, okay? Because micro propagation, it's when you propagate something inside the laboratory condition, inside a test tube or a petri dish, right? So you are culturing something in a small scale, right, in your controlled environment. So let me just draw. Okay, suppose, okay, let us okay, let me just draw a parent plant. Okay, suppose this is the plant and these are the leaves. So you took the leaf, you took a piece and you cultured it in the test tube. No, then it grew into the new plant. Okay, it grew into the new plant. Now this one, let's say it carries gene 3n, okay, 3n, let's say a species of a species 1, okay. Then this new plant also which you generated, it's the exact clone, it's the exact copy of your parent, okay. Means this is also 3n species 1. Are you getting? Okay, let me just make it more clear by giving another example. Okay, take you for example. This is you, okay. You are the student, okay. Okay, you are a daughter. Now you tell me, are you a clone of your mom or your dad? Are you a clone of your mom or your dad? Your dad is Y, let's say, okay. He's having a Y chromosome, no, XY chromosome. Wait, I have to draw well. This is you, this is your dad, and this is your mom, okay. Oops. This one is your mom, okay. And you are daughter, you are student, okay, S, okay. You have half the genes of dad, half the genes of mom, okay. So you are not a clone of mom, you are not MM or DD, okay, you are not DD or MM. See, your dad is, okay, had a homozygous DD for dad, okay, and your mom is homozygous MM for mom, but you are heterozygous D and M. So you are not a clone, you are a new individual, okay, which has arisen out of reproduction, okay. But for clonal propagation or micro propagation, it's like this is your dad DD. I took one cell from your dad and then I cloned, okay, I cloned and I got a new baby, okay. This is the new baby in the test tube. This baby is also DD because it is a clone of your dad. It will grow up to look like your dad, it will behave like your dad, it will have fingerprints of your dad, it will have DNA of your dad. So it is a clone of the original parent. Are you getting it now? So am I not culturing this cell inside a small test tube? Am I not micro-propagating it? So if you see the word micro-propagation of meristem culture or micro-propagation of shoot tip culture, so on and so forth, don't panic. Don't like say, oh my God, it's not in syllabus. What did they ask me? Don't panic, okay? Because it means the same thing. So I think it's clear now. Okay, so let's move on and see what they have written. Now see, all this introduction you don't have to write because here I have included it so that it is what you say, it is clear, your concept is clear, no? Because you are still, this is still new for you, no? So you have to base You have to have a good base, okay? So plants, we can propagate them through sexual as well as asexual means, okay? But clonal propagation is not your sexual reproduction, okay? It's not by fertilization, okay? It is a sexual reproduction, okay? A piece of a non-reproductive part, like a shoot, a root, a stem, a leaf, is taken and is multiplied and grown, and it has the same genetic identity of the parent plant. Okay, so here it is written, no, it's genetically identical copy of the individual plant. Okay. Okay, what else? Blah blah blah. All this sexual reproduction you already know. So you know what is sexual reproduction. You know what's asexual. I've just given you this so that it will your concept is better. So these are the advantages of asexual or vegetative reproduction. Okay, it's faster, no? You can reproduce faster because you don't have to depend on whether the pollen will land, whether the air current is right, whether the temperature is favorable for germination of pollen tube. So many things. Okay, there are so much advantages. So plants, they prefer to do asexual reproduction, like your orchids, no? When you see they sell in the market, they sell those bulbs. Those are the vegetative part, okay? Bulb doesn't mean that it came by reproduction. It's asexual reproduction, okay? It's a vegetative part. Okay, so in vitro clonal propagation. So which means in the lab, let's see. It is a expensive method, okay? In vivo propagation is tedious, expensive, and unsuccessful. Okay, so if you leave them in nature, they may or may not, okay? It may or may not work. No, you might have to wait for millions of years till you get the right orchid that you want, okay? So you go for the lab, okay? You can do it fast, you can do it the way you want it, the way you like it, and at the time you want it. So in vitro propagation through tissue culture, we can call it as micropropagation, okay? So first started by Morel, blah, blah, blah. You read that on your own. So let's look at the technique of clonal propagation or micropropagation. Okay, it involves 3 stages. So better we just look at this. The stage 0 is when you select. Then you initiate and you create the culture. Then you multiply. And then you germinate the somatic embryo. Okay, somatic embryo, I already told you what it is. Then you transfer those young plantlets to the greenhouse, and then from the greenhouse to the field. Okay, so this is the same thing again and again. It's like plant tissue culture, only again and again. So I don't have to read you all this. The note is there. It's just a waste of time because it's just plant tissue culture only. Okay, let's go on. So you can perform micropropagation by axillary buds, by adventitious roots, by organogenesis, by somatic embryogenesis. Okay, so all this we can use all these techniques to create new clones. Okay, somatic embryogenesis, you remember, right? So organogenesis also you remember from an organ. Okay, like a leaf is an organ. You generate the whole plant. Okay. Somatic embryogenesis, from the callus, you create an artificial embryo and get the new plant. Okay. So let's look at the one that they want us to concentrate on, the buds, meristem, and shoot tips. Okay. So by axillary buds and apical shoots. So I have told you many times what's the meristem. Okay. So if you have forgotten, just Google. Okay. So actively dividing meristem are present at the axillary and apical shoots. Okay, so they're present at the shoot tips. So these buds, they can develop into shoots. Okay. So in nature, there are very few axillary meristems, okay, which have the capacity to form the shoot. So by doing it in the lab, you can induce in vitro multiplication And you can develop plants from the meristem and shoot tip cultures and from bud cultures. Okay, so bud and shoot tip and meristem, these all have your meristematic cells. Okay, so meristem and shoot tip culture. So these meristems, apical meristem, it is a dome of tissue. You remember from your diagram, no? You used to draw like that. Okay. It's located at the tip of a shoot. Okay. Then it's the shoot apex. From the shoot apex, you can develop disease-free plants. Okay, then how do you do it? You isolate the stem, the meristem, and then from this stem by a V-shaped cut. Okay, so you have to cut in a V shape. Why? Because the meristem is like that, so you have to cut like this. Okay, then the size of the tip is critical. Okay, so you have to be technically you have to be good in cutting. So if you have a larger explant, okay, you try to get a bigger a culture, a bigger plant part, your chance will be better for survival, okay, because the cells will not be damaged. So if you want to succeed, you have to take from the actively growing shoot tips, okay, so you have to choose, okay, which look yellowish or which look old. Don't choose, you choose the young and growing ones. So you culture it in a medium, MS medium. And then let's look at the diagram. So here, this is the shoot apex, okay. Then they cut it like that in a V shape. Then they put it in this culture, okay. This is the shoot tip. Then you can see that the shoot is developing, okay. Small leaves are growing, small branches are growing. Then you can see that the roots also will grow, and then you're getting the new plant, and then you take it to the environment and you pot it, okay. So in the first stage, when you put it in the culture medium, in the MS medium, you have to give your hormones, no? You go for your cytokinins and your auxins. Then if you want to have a lot of shoots, you have to give high level of cytokinin. Then after you got the shoots, then you change the concentration, you give more auxin, and then you get the roots. Then you transfer it, okay. You can read on your own, no girls, it's the same thing again and again. Okay, looking at bud culture, plant buds, they have this active meristem. Okay, so there can be two buds, single node culture and axillary bud culture. Okay, single node culture is not there in the syllabus, so I didn't include it. Axillary bud culture is there. So buds also, no, like flower buds and all, they have these meristematic cells, okay, actively dividing cells. Okay, so again here, simply you just take a shoot tip which has a bud, okay. Then you add cytokinin. Then if you add cytokinin, apical dominant stop and axillary buds develop. So what this line means is that, see, this is apical dominance, means this plant keep growing upwards, okay. Then what happen if you take a pair of scissors, okay, shears or scissor, whatever you call them, and then you trim this bud, okay, you trim it. So what happened when you trim it, no, instead of keep on growing upstairs, it will develop shoots from the left and the right, okay. So these are your axillary buds, okay. Just like a rose, no, if you don't trim your rose bush, no, it will keep on growing upwards. What happened? Okay, where am I? Okay, so directly to the diagram then. Like a rose bush, no, if you trim your rose bush, it will become short, but it will develop a lot of rose flowers from the sides, okay. So this is a plant You culture it, you develop the shoot by adding cytokinin, develop the buds by adding auxins. Okay. Again, these images are almost, it's like a repetition. All you have to do is change which explant. Sometime they ask for meristem, you took meristem explant. They ask for auxiliary bud, you took auxiliary bud explant. Okay, shoot tip explant. Okay, so that everything else is same, just the beginning, the explant part. Okay, then let's look at shoot tip culture. So shoot tip, it is when you remove the tips of the shoots. Okay, and in the shoot tip there is those meristematic cells. Okay, so that's why they say shoot tip and meristem. So you culture it on your agar, your medium, or your liquid medium, or MS media. Okay, then you add those auxin, cytokinin, etc. Okay, let's look directly at the protocol. This principle you can read it on your own. It's not tough. So you remove the young twigs, then you sterilize, then you transfer the explant to petri dish. Then you remove the unnecessary part. Okay. Then remove all the outer leaves. Okay. Then the apex is exposed. Cut off the apex with scalpel and transfer those to the agar medium. Okay. So the steps you go through on your own. Okay. So just here, simply they took the shoot tip. Okay. They took the shoot tip, they cleaned all the, they removed all the unnecessary plant parts, then they transferred it, and again they added the auxin, cytokinin. Then after you get the plantlet, they take it to the greenhouse, and so on and so forth. Okay, so all the methods are the same. So these are the applications of micropropagation, means the advantages. Okay, so whether they ask you for shoot tip or root tip or meristem or whatever, the answer is the same. Let me repeat. If they ask you for the advantages of auxiliary bud, you will give these only. Okay, you will give these only. High rate of plant production, disease-free plants, seeds, cost-effective, so on and so forth. Okay. Whether they ask you about meristem culture, advantages are the same because it's basically micropropagation, no? Similarly, even the disadvantage is the same. Whatever they ask you, whether it's shoot tip, what is the disadvantage? You will tell them this only, okay, these only. Contamination problems can arise, brewing medium, vitrification means when they become frozen, no? Then high cost, right? Okay, so with this topic is done, and then I'll see you again on Saturday with a new topic.